Assalamualaikum warahmatullahi wabarakatuh. Today we're going to present about 2D Sds Page. What is 2D Sds Page? 2D Sds Page is a technique used to separate protein from complex mixture based on two properties, which is first isoelectric point and second is molecular weight. This high resolution method allows for the detection of protein isoforms and post-translational modifications, which are crucial for understanding the proteome. The proteome represents the complete set of expressed protein in a cell or organism at a specific time and also under defined conditions. Since protein can exist in multiple isoforms due to the post-translational modifications or PTM, the proteome reflects this diversity. So, studying the proteome through techniques like this 2D Sds Page will provide insights into protein function, interaction, and also disease mechanism. And so moving on, 2D Sds Page is essential for proteomic studies, allowing for protein identification, comparison, and also structural analysis, thus advancing our understanding of cellular biology and also disease pathway.
Now, let's move on to the protocol for 2D gel electrophoresis in separating proteins. There are 4 steps, which are sample preparation, first dimension separation by isoelectric focusing or IEF, second dimension separation by gel electrophoresis, and staining.
Let's take a look at the first step, which is sample preparation. First thing first, we need to ensure that the protein concentration is being standardized for all samples. To do this, we can use standard assays like Bradford assay or BCA assay to measure the concentration of proteins in a solution. Next, the protein will be reduced by using reducing agents to break disulfide bonds in the proteins, where this step makes sure that proteins are in a linear form, which is necessary for accurate migration during the IEF. Then, the sample will be mixed with the carrier ampholytes that are already on the immobilized pH gradient strip or IPG strip to assist proteins in migrating to their specific isoelectric points or PI during the IEF.
The second step is the first dimension separation by IEF. Proteins can be separated by their isoelectric point. The isoelectric point is the pH at which the amino acid has no net charge, which means the positive charge is equal to the negative charge. At their PI, the positive and negative charges balance out, which makes the protein stops moving or migrating in an electric field. So, a pH gradient is used to separate different proteins based on their isoelectric point. And this technique is known as isoelectric focusing. The third step is second dimension separation by gel electrophoresis. Gel electrophoresis further separates proteins based on their size, which is their molecular weight, when an electric current is applied. Sonagra and Dolaria 2022 stated that negatively charged molecules move toward the positive electrode, which is the anode, while the positively charged molecules move toward the negative electrode, which is the cathode. Plus, smaller molecules migrate faster and further through the gel compared to bigger molecules. The fourth step is staining. There are 2 purposes of staining, which are visualization and comparison. As per biored.com 2025, staining is vital because it makes the molecules visible in the gel, allowing their migration to be monitored and documented. After that, staining allows direct comparisons, such as identifying and also determining variations in protein expression.
Now we're moving on to the 3 types of staining methods used in proteomics. First, Comessi blue staining. It is simple, cost-effective, and easy to perform. However, it is the least sensitive with detection limits of 5 nanograms, and it is compatible with mass spectrometry. And the second type of staining is silver nitrate staining. This is the most sensitive method, detecting as little as 0.1 nanograms, and it provides quantitative data, but is time-intensive and not very compatible with mass spectrometry. And the final type of staining that I'm going to explain is fluorescent staining. It offers sensitivity close to silver staining with a detection limits of 0.25 to 1 nanogram, and it provides the best dynamic range for quantitative analysis and is also compatible with mass spectrometry. So what is the advantages of 2D proteomics? First, it offers high resolution, allowing thousands of proteins to be separated simultaneously. This method is also effective for identifying post-translational modification, since such changes can alter the isoelectric point and molecular weight of proteins, causing position shifts on the gel. Additionally, 2D proteomics is highly reproducible. Now, onto the disadvantages. This technique is time-consuming and technically challenging. Automation is difficult and it's less effective at recovering certain proteins, such as membrane proteins. And finally, it has limited usefulness in protein identification because matching spots on gels can be complex.
Assalamualaikum and hello everyone. I'm AUNI and I am going to explain how to identify proteins using tandem mass spectrometry. As you guys can see in this flow chart, following 2D gel electrophoresis, the protein spot of interest will be cut either manually or automatically. If done automatically, we will be using automated spot cutter. After that, the protein spot of interest will undergo protease digestion using trypsin and pepsin, and the dye will also be removed to avoid interference. The extracted digested peptides are now ready for MS. In tandem mass spectrometry, a biomolecule of interest is isolated from a biological sample and it is fragmented into multiple subunits in order to elucidate its composition and sequence through many series. Tandem mass spectrometry begins with an ion source, and as you guys can see in this video, it utilizes MALD, matrix-assisted laser desorption ionization, which converts our sample into ions. And there is also a mass analyzer which separates the ions based on their mass-to-charge ratio. A common mass analyzer, a quadrupole, only allows specific ratio to pass through, while the others will crash into the rods. The specific ratio that has been passed through are called the precursor ion, which is our biomolecule of interest. This ion moves into a collision cell, typically another quadrupole, where energy is applied to them in order to fragment the ions into a predictable pattern. This process is also known as an ion separation. These fragmented ions will now move into a mass analyzer such as a TOF, time of flight, which separates these product ions. These product ions will now move into a detector to generate an MS spectrum. When analyzing an unknown protein, the MS spectrum contains many overlapping fragments, making it hard to determine the protein sequence. However, each protein has its distinct spectral pattern. Therefore, analysis software will compare the pattern to a non-database sequence.
Now, I will be talking about mass spectrometry or MS. MS is a powerful analytical technique used to measure the mass-to-charge ratio of ion, helping us identify and analyze molecules in various fields. What is mass spectrometry? Mass spectrometry is an analytical technique that determines the mass-to-charge ratio or MZ of individual ions. This allows us to determine molecular mass, structure, and composition with high precision. How does MS work? To measure the mass-to-charge ratio of individual ions, molecule converted to ions in the ionization stage possess net charge, which is negative or positive charge, separated and detected based to MZ ratio. What is MZ ratio? The MZ ratio is the mass of an ion divided by its charge. Mass spectrometry measures this ratio to identify molecules. Example of a peptide fragment. Let's take the peptide curve as an example. Its neutral form has a mass of 1059 daltons. Single protonation plus one hydrogen charge. When a proton hydrogen is added, the mass increases to 1060 dalton. Since the charge is plus 1, the MZ ratio is 1060 over 1. Double protonation plus 2 hydrogen charge. If another proton is added, the mass increases to 1061 dalton. Now the charge is plus 2, so the MZ ratio is 1061 over 2. In mass spectrometry, adding charge increases the mass, higher charge results in a lower MZ value. This helps in identify and analyze molecules more efficiently.